Yeah, so I'm really excited to be here, and I'm going to be talking about a study which is really tracking the emergence of memory representations across an extended time period. So it fits very well with this theme of our conference. So there's no need to introduce the hippocampus and medial temporal lobe learning system in this crowd. So it's thought that unique patterns of activity in the hippocampus are laid down when particular events are initially encoded into memory. So here's an example of a nice pre-pandemic event, visiting a famous museum and traveling. However, it's known from a large body of data that over time, our memory representations become more distributed across hippocampal cortical networks through a process that we refer to as systems level memory consolidation, referring to this reorganization of our memories over time across hippocampal cortical networks, such that when we later retrieve representations from the past, these are associated with more distributed patterns of activity across the hippocampus and cortex. And a key focus of my research is to understand post-encoding mechanisms that support memory retention. For example, to better understand the different kinds of brain states that occur after encoding and support memory consolidation. But since I won't actually be talking about this topic today, I just want to briefly advertise a paper that was published last year demonstrating the causal influence of awake post-encoding processes that occur during an immediate post-encoding time period that actually causally contribute to subsequent memory retention. And this is a little bit analogous to some of the approaches that we heard about yesterday for manipulating post-encoding processes in animal models. So instead, today I'm going to be really zooming out in terms of the timeline of memories, similar to what we heard from Carrie Hoffman's talk. And I'm going to be asking the question of how do our memory representations change over the course of time? And specifically, can we longitudinally track these changes across an extended learning period in the human brain? So multiple theoretical and computational accounts of memory consolidation posit that this process of systems level reorganization is important for aiding in the extraction of structured knowledge across our individual experiences, or the extraction of statistical regularities across individual events that we've experienced. So again, multiple frameworks, including the complementary learning systems theory, emphasizes this idea, as well as multiple trace theory, as well as the trace transformation theory. So this is an important feature of consolidation. And along these lines, the inference of associative relationships improves over the course of time and sleep. So the key idea here is that over time, cortical memory representations are likely reflecting structured information across our individual experiences. And here's a really nice example of this from prior work from Alexa Tampari and Leila Devachi. I think I saw Alexa in the event before. And so in this study, object scene pairs were encoded by participants such that there was some of these scenes were repeated across individual object scene associations. And this allows for a structured representation to emerge over time that might link these objects, which are paired with the same scene across individual associations. And this pattern was actually found in terms of brain activity. So when participants were scanned 1 week after learning these associations, they found that patterns of activation in the medial prefrontal cortex were more similar between objects that were paired with the same overlapping scene compared to objects that were paired with different scenes, suggesting that this structured representation emerges across 1 week of post-encoding time. And this was also found in the hippocampus as well. But one question that comes up in really nice studies like these is, what is the timescale of the changes that we expect to see? So clearly these can be captured at relatively short timescales, such as overnight or, at 1 week. But we really think that these changes should be emerging over a more gradual time period. And so in this work, we wanted to address this question: how are novel stimulus representations altered as a function of repeated structured exposure, and we wanted to do this across an extended time scale of months. So to do this, we performed extensive fMRI scanning on a small sample of individual humans, piggybacking really on recent work using this kind of an approach of extensively sampling individuals over time. And in particular, what we did here is we performed repeated fMRI scanning, which is shown in each of these purple dots, on 3 different participants over the course of about 4 months of the study, in conjunction with behavioral training in the form of a serial reaction time task, which I'll go over in a moment, as well as a working memory task, which I won't be talking about today. And because it's challenging to perform these kinds of studies on naive participants, you don't want them to drop out in the middle of the study. The 3 participants in this study were myself, as well as my 2 collaborators on this project, Anastasia Kianaga and Jacob Miller. Here we are posing outside of the MRI scanner, wearing these custom-made head cases that we got for this project. And I just want to thank Mark Desposito for allowing us to kind of conduct this crazy study and collect all of this data just before a number of us left the lab. So in order to track memories over time, each participant was repeatedly exposed to a unique set of these novel fractal images that you can see here. And in the first phase of the study, we simply learned stimulus response mapping. So we learned that we had to press one of 4 different buttons in response to seeing each of these fractal images. And this took place during the first 2 sessions in the study. And in the remainder, most of the experiment that was remaining, participants were then exposed to structured regularities in the form of these temporal sequences that you can see here. So what's happening during the serial reaction time task is participants are simply viewing a stream of images one after another on the computer screen and making their button responses, but these temporal sequences are embedded within the stream of images. And there were a total of 4 sequences that participants were exposed to, each containing 4 images, along with these non-sequence images, which were interspersed throughout the stream of stimuli. And we chose this kind of task for a few reasons. One being that it provides a behavioral measure of sequence learning. That is, we should see reductions in response time for items 2 through 4 within each temporal sequence over the course of learning. And here I'm showing you behavioral data from one subject, just so you can see what this looks like. What we see is a really clear reduction in response time to items 2 through 4 within the sequence over the course of 80 days of training. Hopefully you can see my whole screen, which I can't. Okay. Just to give a shout out to Aaron Bornstein, one of our great co-hosts for this conference. I'm actually borrowing analysis approach that he developed from a prior study here. And so we also use this kind of task because it allows us to extract different neural measures of structured memory representations over the course of time. So I'll get to this in a second. So in order to actually track memories over the course of time in this experiment, what we did is during each scanning session, participants viewed each individual fractal stimulus in isolation, a total of 8 times within each individual fMRI sessions. So this allows us to ask a number of different questions, such as how does overall activation to these fractals change over the course of this experiment? We can also ask how are multivoxel patterns as a proxy for representations actually shaped by these sort of structured experiences that participants are having. And before getting to these questions, I just want to briefly address how we've been thinking about modeling changes in brain activity that we see over time in this task. This is an important question to consider because if we want to be able to actually discover where in the brain changes are occurring over time, we need to create a model of what those changes should actually look like. So a simple approach would be just to assess where there are changes that are occurring linearly over the course of time in this experiment. And I've analyzed the data in this way previously, although I won't be showing that here. And this approach is a little bit unsatisfying because it's possible that changes may be occurring kind of at any point over the course of 80 days in this experiment. So it might be really incorrect to assume that these changes are occurring in a constant manner over the course of time. So to get around this issue, we instead fit these kinds of nonlinear sigmoid functions to the data that look like this. And this approach is nice because it allows us to actually parameterize changes over time, both in terms of when the changes might be occurring. We can look at the inflection point in these curves, as well as the rate of change. So for example, we can use these curves to detect changes in activity that might occur very early on in the experiment, middle of the experiment, or very late in the experiment. And they can also detect changes that might occur more gradually over the course of time, more similar to a linear kind of change. So I think it's a nice approach because it gives us kind of the best of both worlds in terms of being able to flexibly detect different patterns in terms of changes over time without, fitting something like a 10th order polynomial function, which might describe the data well, but might be difficult to actually interpret. So going back to our questions, first, how does fractal evoked activation change over the course of time? Here we specifically wanted to focus in on the medial temporal lobe, so the hippocampus and medial temporal lobe cortex here, and ask questions such as, does hippocampal activation potentially decline over the course of the experiment? So for example, if hippocampal representations are present initially, but maybe these memories kind of migrate to the cortex over time, we might expect to see this decline in responses over time, which have been reported in a number of neuroimaging studies before. On the flip side, we would expect to see that cortical activation would likely increase over the course of time. So to get at this question, we asked whether each individual voxel within a given brain region of interest shows systematic changes over time, and asked whether the total proportion of voxels that we see within a given brain region actually shows a significant increase or a significant proportion of voxels that show a decrease over the course of time. And here I'm showing the data for regions within the medial temporal lobe. And what we see here in gray are these null distributions of changes that we'd expect to see just by chance based on the number of voxels that we're looking at. And here the dots that are colored in red are actually showing where we're seeing significant either increases or decreases in activity over the course of time. So we can see that actually around 20% of voxels in the hippocampus are showing an increase in activation over time, and similar proportions for the entorhinal and perirhinal cortices. And we're only seeing a decrease here in the parahippocampal cortex over the course of time. So this suggests that memory representations may be developing in these regions, but this is really an indirect proxy for the development of these representations. There may be other cognitive processes that are changing over time and being reflected in overall levels of activation that we're measuring here. So to get this more directly, here we wanted to test the idea that shared representations of items within an individual sequence, similar to pair coding neurons, may emerge over the course of time. And more specifically, we predict that items that are part of the same temporal sequence would develop or come to elicit a more similar pattern of activation over the course of the experiment. And the way we parameterize this is by representing this in the form of a correlation matrix here. So we're specifically asking whether items that are part of the same temporal sequence become more similar to each other over the course of time, and that this would occur for each of our 4 sequences, that these patterns may be distinct across individual sequences as represented by blue in this matrix, so a decrease in similarity over the course of time. So first, focusing on the hippocampus, on the y-axis here, I'm showing an estimate of model fit, or how strongly this sequence representation is present in the data for each individual session and participant over the course of time in this experiment. And here what we can see, I think pretty clearly, is evidence for this kind of sequence representation emerging over the course of time in the experiment. We can see that these values begin at 0 and go up over time. There's clearly some variable expression of these representations across, over time across individual participants. What I think is pretty striking is that there's this increase over time, and it remains relatively stable, such that at the end of the experiment, we're still seeing evidence for these sequence representations in the hippocampus after 80 days of training on the same task. So we find evidence that this shared sequence representation emerges and persists over time in the hippocampus. We next turn to the medial temporal lobe cortex and perform the same analysis. And when we perform this analysis at the same level of granularity, so looking within a given region of interest, do we see consistent effects or development of representations across our participants? We don't actually see a consistent effect across participants, sort of across whole regions in the medial temporal lobe cortex. So we then turn to asking whether there might be more localized sequence representations that are present and potentially restricted to different portions of medial temporal lobe cortex. And so we did this using a multivoxel searchlight approach for the fMRI folks that are here. And really what we're asking is, do we see evidence that these sequence representations are emerging over time in the experiment? And we do find some evidence for more localized representations being developed. Here I'm showing the results of where we're seeing significant effects for 3 participants in the data set. So subjects 1 and subjects 3 are showing sequence representations being developed in posterior parahippocampal cortex, whereas subject 2 is showing these representations developing in perirhinal cortex, where we'd expect to see these responses based on the notion of paired coding neurons, which have been extensively found here. And here I'm showing what the time course of these changes look like across each of our 3 participants. So we can see that there's a bit of variability in terms of when in time these effects are emerging. One question that we can ask now, since we've characterized these changes in the hippocampus as well as the medial temporal lobe cortex, is does hippocampal learning actually precede the emergence of these representations in the cortex? So what I'm showing here is simply overlaying changes in representations in the hippocampus for these 3 participants versus the changes in their representations in medial temporal lobe cortex. I apologize, the graphs are a little bit noisy, there's a lot going on, but what you can see is if you focus on these regression lines is that at least for subjects 1 and subjects 3, we see a pretty clear trend such that representations are emerging earlier in time in the hippocampus as compared to the medial temporal lobe cortex, maybe also a bit for this participant here. This is consistent with the idea that these hippocampal representations tend to emerge earlier in time than cortical representations, consistent with the idea that the hippocampus is more of a fast learning system. So you might be wondering, well, what else is happening in the brain? There's a lot more than the medial temporal lobe, which is involved in long-term memory representations. And so we wanted to go back to our first approach and ask whether we can see increases in activation to fractals over time in the cortex. And here we're performing this analysis within individual regions of interest in the cortex from this parcellation. We do find that many regions throughout the cortex that we would expect to see do actually show an increase in activation over time. So we find this kind of an effect broadly throughout the medial prefrontal cortex, medial parietal cortex, the anterior temporal cortex, as well as ventrolateral prefrontal cortex, and the temporoparietal junction. So these are regions that we would really expect to see these changes in. And we next want to ask whether we see evidence for the emergence of these structured sequence representations developing over time using our multivoxel searchlight approach. Specifically, we're asking whether we see shared sequence representations emerge over time here. And what I'm showing is results for our 3 participants in terms of where we're seeing these sequence representations emerge. And whoops, I'm starting to run out of time. For our first subject, we see that these representations are emerging in medial parietal cortex as well as lateral temporal cortex. Subject 3 is showing some similar representations developing in medial parietal cortex, medial prefrontal cortex, as well as the temporoparietal junction. Surprisingly, we don't see consistent effects across all of our participants, and clearly the location of these effects are varying across participants as well. So given this variability, we next wanted to ask whether we might see shared representations for different kinds of structures in memory in this experiment. So we're specifically asking, do we see shared representations based on stimulus categories, maybe reflecting a coarser structure in the experiment? So asking whether there are shared representations across items that are part of sequences in general and part of non-sequences in general, that these might be distinct across these categories. So very quickly, when we do this analysis, we do find for, again, a subset of our participants that we see these categorical memory representations developing over the course of time. So for subject 1, we see kind of relatively widespread evidence for this categorical representation. We now see this for our second subject, and we don't see this for a third subject. And since I'm short on time, I'll kind of skip over this part, but we actually find, we're able to find converging evidence from behavior suggesting that these two participants actually have this kind of categorical structure and are expressing this through how they're responding in the serial reaction time task. So sorry for taking a bit too long here. Just to wrap up quickly, I've shown you evidence for the emergence of memory representations that reflect task structure over the course of months. We see enduring hippocampal involvement over the course of months. That hippocampal representation seems to be preceding those in medial temporal lobe cortex over time. And we're kind of seeing a multiplexing, or a lot of variability in terms of cortical representations differing across participants, different levels of granularity in different brain regions. So I think I have the chicken and I've gone on too long. I just want to quickly say that this is a really rich data set. We're working on a number of different analyses on it. And eventually, we're planning to release the data and publicly share it so that others can hopefully learn from it too. So thank you for your attention. Thanks to my collaborators on this project, as well as Luke in my lab here at UCI, who's been helpful in terms of contributing to these analyses that I showed.